Hey guys, welcome to our channel. Hope everyone's having a good new year. And I know I certainly am trying to, watching some football, cooking some ribs, and trying to get through our Bible section of Bible changes that we all agreed on, or those of it, or we agreed on. Um, and Megan helped us out there. And we've already gotten our letters to go through all the categories, 7 of them. Actually 8 if you want to call translations, which kind of proved to be a bit much in this case. But um, we've got our numbers. I'm going to show them to you. I'm going to provide a link where we extracted the numbers or where we converted the numbers. And this has seemingly become slightly more difficult since um, having a talk uh, with my cousin, who is super intelligent and was a major of quantum physics. And we've been trying to, honestly, I've been trying to throw stuff out, trying to be more scrutinizing, and in doing that it becomes difficult because what do you throw out? It's all here in the changes. And no matter how many random letter sequences I run, some go through, some don't. Some don't present any sort of a pattern at all, even if they do go through, and some of them do. It's kind of all over the place, and different categories don't go through. Uh, the random letters are kind of random. Um, that's what I'm finding out. But now I'm trying to scrutinize our changes more because of all the time we're spending on these. We know we have another data set, the variance percentages, and we know we have those charts that are good data, and we went a couple directions and got a couple data sets, and this is a major data set. This is what's in the changes. It's there, and but I find myself scrutinizing them more and trying to throw stuff out, trying to see if they're trying to make things not go by on the changes. Why would I do that? Well, if we're wrong, then we'd like to get this thing turned around and going in the right direction. But it's looking a whole lot like we're onto something because the random letters just aren't the same. And but I guess the point I'm trying to make is these have become more difficult to figure out. I always write something down, especially in these numbers, as a frame of reference. Um, but there's a ton of stuff in the numbers, and some of it can be thrown out. But what? Maybe when we get a better understanding of what's going on, we'll have a better understanding of what to throw out. Um, but here are your number. I'm not even going to read them, guys. I'm going to save the time. I'm going to provide a link. Here they are, corresponded to their letters that we got off of a numerology chart you can find online. And it goes all the way to the end and then comes back. And you can see, if you want to pause it, you can definitely see the letters and the numbers that correspond. Now, we've already come forwards and backwards through finance, and we've went forwards through genetics. And by the way, I think I mentioned we got all the letters to go through all the categories, which are finance or stock market, um, genetics, medical, chemical, technology, and then research groups, random hits, which we're looking for other categories to surface or anything different that didn't come up in our other searches, and translations. All right. And now our numbers have come through finance and now have come through genetics. And by the way, I made a mistake on this and got a couple numbers off again. And they were still going through in blocks of 4. So I got them to go through in blocks of 4 in a couple different ways, one by mistake. Um, so interesting, because a lot of times that does not work with the shorter codes. But why would that be? Why would I have to drop back to 3 numbers in the shorter codes in order to make something work? Why wouldn't the 4 numbers just work like they do here all the way through? Well, that's a good question. And maybe the answer is a shorter code. Maybe it was run different. But yeah, we're learning a lot. We're getting places with this. So anyway, guys, first of all, now we're coming back. We already went forwards in genetics, but now we're coming back. You got 3226, which ends up being HOXC10 homeobox humans, and it's a gene ID 3226. All right. Well, that's not all you got. You got population level purifying selection and gene expression, and that's pages in some sort of a publication. I would like to throw that out, but I don't know what's within those pages. So it's really difficult to throw it out. You got common variant mapping to CACNA1A. And there's your number. Variant mapping. Well, we got all sorts of variants, right? You've also got strain RB2374 genotype CNC2OK3226. And there's your 3226. So like literally a host of stuff. Here's genetic analysis of wheat and molecular marker assisted breeding. And there's the number. Hard to throw that out. And here's human vascular disorder. And there's the number. It's tough to throw that out. So that's a page of stuff, and it's tough to throw out. And I will provide the search guys, and it literally will go on for pages. And I guess you guys can decide for yourself what's relevant and what should be thrown out. And if you have ideas of what to throw out, let us know. But what I wrote down was the HOXC10 gene. All right. And so if you move on from that, you have 2159. And 2159 is long intergenic non-protein coding or non-protein RNA. LINC 02159. Plus, you got localization of a gene for non-syndromic hearing loss. And there's our number, 2159 down there. Hard to throw that out. There's your non-protein coding RNA again. Definitely can't throw that one out because we get those all the time. You got some stuff in here for PubMed, 5 structural variants of Pastrel. You got another strain, RB1715, and genotype AQP2OK2159. So there's your 2159, and it's a strain, and I'm not sure those things. Some of those things are alleles, and some of those things are alleles, or whatever, however you pronounce those, and some of them end up being viruses. But you got a couple of those going on here. It looks like RB2159, genotype INS. So 16 OK2919. So there's RB2159. You got some other stuff here. You got complex coding and regulatory polymorphisms. And there is 2159 coding, so it's hard to throw that one out. You got a fly base reference report. Once again, 2159. and we can't throw that out because the animal stuff was predicted. So again, there's a page of stuff we can't, that I really am having a hard time deciding if we can throw any of it out. But because it's all there, it's all within these changes, and without a better understanding of exactly what they're doing, it's all relevant, seemingly. But maybe we'll get better at this whole thing. So, but if you move on from that, yeah, and I'll provide these searches, guys. There's stuff that obviously isn't going to make the video here, because I can't read pages of stuff. But if you move on from that, you have 2595. And 2595 ends up being SSR 2595 gene. And you also get non-protein coding RNA 2595. You also have strain RB2595. That's another one of those strains. And yet another strain, VC2595. You also have Epilepsy, a comprehensive textbook. And you have 2595 genetics right there. And do I see my number? Oh yes, 259 VT2595. Carnioer habitus genetics center. It's a genetics center. View on worm-based species. So there's more of your animal stuff that my fiance predicted. You've also got GANC, glucosidase alpha neutral C, Atlas of Genetics. And there's your 2595. We could probably, that one, I might throw that one out and not use it. If it was the only thing we had, I might mention it, but I might say we can throw that out. I don't know, guys. Even if we could throw it out, there's so much here that we can't throw out that it's as a possible thing to be calculated that it's how about preconception and prenatal genetic screening? And there's 2595. So yeah, you got a few things going on with 2595. So if you move on from that, and I wrote down SSR 2595 gene. So so if you move on from that, you have 8336, and that ends up being data set Umpqua salmon genetic pedigree. Oh, well, why would I be worried about salmon genetic pedigree? Well, my fiance predicted animal stuff. And when she predicted it and I see it, then I have to include it. ID 8336. So there's your salmon genetic pedigree. You also have a fly base report, CG 8336. You got something. You got a H2A1 protein gene 8336. And so you also have one of these variants, NM underscore 001267550.2 TNN, or TTN, excuse me, colon C25008C, greater than TPCYS8336. Those are so long I hate reading them, but single nucleotide variant. You got a couple of those. So you actually have identification of a gene that positively regulates expression of Mutant. So, gene mutation stuff going on here, and there is the number. Hard to throw that out. So once again, you're just left with pages of stuff that absolutely could be calculated in these numbers. But if you move on from that, and what I took away from it was the salmon thing, because my fiance predicted animals stuff coming up here. So that's interesting to me. If you move on from that, you have 3241, and that ends up being HOXB4 Homeo Box B4, and it's gene ID 3214. So you got a gene ID of the Homeo Box family and codes a nuclear protein, so it's a protein coding gene. You also have 21UR3214 gene, and that is worm base. So there's some more of the animal stuff. You also have a strain, MT3214, genotype, SEM2N1343, and that's a strain. And do I see my number? I see my number. You got you got that genetics center again. Carnahabditis. I don't know. Canorhabditis Genetics Center. So, that came up before just a minute ago. You've also got another strain, CB3214. Do I see my number here? If I don't see my number, we're moving on from that. I do not see my number, so we're moving on from that. I do see a targeted gene disruption of the endogenous genus CABL locus. So, gene disruption. All right. Hard to throw that out, too. Like, it's tough to throw any of this out. So, that's your 3214, and again, we pre-designated blocks of 4 or sequences of 4 for this, and if they didn't go through, we were going to make note of that. Instead, they went through in a couple different ways, in sequences of 4. So, actually, that was 3214, and if you move on from that, you have 9241, which ends up being NOG noggin gene. Gene ID 9241. All right, 9241. And I don't think this one was a protein coding. But either way, you got that gene, NOG noggin gene. Oh, look. Where's my number here? 9241. How about sheep genetics information? Sheep genetics. Animal. Oh, lion and the lamb, right? Wolf and the lamb, sheep. The sheeple. But sheep genetics information, definitely can't throw that out. There's more of your NOG gene. You got, do I see my number? 9241. Yeah, you have orchid, ORC ID. Connect, and you got some research, genetic research there, non-genetic variation. You got some more ORCID, studying gene expression in yeast, and expression and contribution of, expression and contribution of virulence of each polysaccharide, G9241. So yeah, you got a page, and it goes on, but that's all there. Sheep genetics information, actually, and looking at it again, is what I would take from that, and the noggin gene for humans. because there's more of that animal stuff in the National Wildlife Federation. I said fund yesterday, but it's federation. All right. So, and we're going to have a lot of circled entities to run these numbers against. So, and see who may be using the quantum network here. This one ought to be interesting for those who are following along and want to know what's going on with their Bible changes. I think we're coming a long ways in finding out. But if you move on from that, you have 2159, and that takes you to, and I wrote it down because we've had it before, it takes you to the Jackson Laboratory. Allele, type, gene symbol, gene name, and all sorts of stuff. And my number is right here. B2B2159 CLO. J, this mutation. So it's a mutation. They're studying at the Jackson Laboratory. We've had them before, not in this search, I don't think, but we've had Jackson Laboratory in previous searches. So I thought that was interesting. You also have another strain. 2159. So you have strain RB71 or 1715 genotype APQ2OK 2159. So there's your 2159. You have another strain with genotype 215, another strain RB2159, excuse me. So you got two strains, you got three strains, KW2159. You got that main thing again. Do I see my number? 2159C. Yeah, a carrier may request but not require that an individual will undergo genetic test if the request is made pursuant to research that compiles with common rule. Yeah. All right. Complex coding and regulatory polymorphosis. And there's your 2159. We actually got that a little earlier. Involvement of mouse Rev3 in tolerance of endogenous. And in cancer genetics, the paradigm holds the sequential mutation of a series of oncode genes and tumor suppressor genes. So that's interesting. You got cancer and animal genetics right there. Mouse. So that's 2159, and I'll provide these searches, guys. We'll try to get through these a little faster. If you move on from that, you have 1711. And 1711 ends up being a non-protein coding RNA, LINC01711. RNA 1711, non-protein coding. So you also have engineering the Drosophila genome, chromosome rearrangements. So in my numbers there, 1711, can't throw that out. Bacterial artificial chromosome-based physical map. Rice genetics map. We've had that before, guys. The rice genetics. So it might have been. What was the rice genetics? I might not have wrote it down, guys. But we had rice genetics in our last search as well. You also have strain VC1233 genotype OCR-2OK1711. So there's your 1711. You also have VC1711. That's another strain. SPBC1711.13. That's a gene. Well, yeah, hard to throw that out, but that's one that I can't really throw it out, guys. Like, I don't know. I want to throw some of these out just to be more stringent, but then I think about it. I don't know exactly what they're calculating. I don't know what calculations they're making, so it's hard to know what to throw out until we knew more. Genetic information. So title 222-1711-C, confidentiality of healthcare information. Go down here and read. It says including individual cells or their components or genetic information. You also have FAM 171A1 gene, F1711 protein. So you got a protein coding gene. So you got a few things going on. The next thing, oh, I do see my number. And they're talking about epidermidis genetic clusters. So you got it, and again, you got a few pages of that. I'll provide the links. That was 1711. If you move on from that, you got 1171. And that ends up being identical mutations in CB or in CBS gene. Let's take a look. Okay, well, identical mutations in CSB gene. Excuse me, I wrote that down wrong apparently. So there's mutations, which we've had before. You got the language of genetic interaction, and there's your number, 1171. That's tough to throw out. I'm just kind of skimming through here, guys. You got another strain, RB1171. You got another strain, LD1171. You got a college course, which I will throw out because there's so much other stuff here. We can throw out the college course. Why not? I'm not so sure we should, though. Those could be important. Who knows what's within that college course to calculate. But, yeah, you got a few things going on here. Yeah, you got some other things that I'm just kind of skipping over. But I'll provide the link. And you guys can decide what you think should be pitched and what you think is relevant. But moving on from that so we can get done, 5285 is what you get next. And 5285, I got test code for Baylor University again. Now, we got several of those in our last search. Medical genetics test details, Baylor College of Medicine. Test code 5285. So there's another test code. Fundamentals of Mammalian Genetics, Department of Biology. Cancer genetics is there in the fundamentals of mammalian genetics. So we've had that before. You got more mammalian genetics. Fly-based gene report, CG5285. You come down here, you got MRRC colon 0429. Do I see my number? Yes. You got MTGXR5285BTLR. I don't know what that may be. Citation clipboard. We can throw that one out. But we can't throw out clinical cancer genetic services, Jefferson University Hospital. And you get a phone number here, 5285. I'd love to throw it out, but because we've got so much cancer stuff, I can't. So you gotta. That may be important, so it's there. We'll mention it. But that's just one page of stuff, guys. If you move on from that, you have 8241. And gene ID 8241, RNA building motif protein 10, RBM10. And this is apparently oh, it is. It's a protein coding gene, guys. So you got a protein coding gene. Wanted to make sure I knew that was a protein coding gene. Binding motif protein 10. You got that. It's the same thing. I avoid Wikipedia and wiki anything. So we blow by that. Course details, AGRO 8241. It's students' experiments in breeding, genetics, genomics, and physiology. Yeah. What do you guys think? Throw it or keep it? As data. Study materials. So, BS5 is work. Do I see my number? I do. 824. Pepper BS5 SOL Genomics Network. So that could definitely be important. So you got a few things going on with even that. So if you move on from that, you got 5111 and 5111. I wrote down SLR511 gene, and it is a protein coding gene. But you also get other things. I think we've actually got this before. Oh no. Well, yeah, we have got staff at the Yale School of Medicine, but now you have Andrew XIAO PhD, genetics, Yale School of Medicine. So he's a professor in the Department of Genetics. Oh, Stem Cell Center. He's a member of Yale Stem Cell Center. That seems important. You also have another Alan Luing, PhD, and research and scientist in genetics. So you got a couple of research people in genetics that this number goes to. Coding and analysis of RNC ERA REC00PERON. So you got coding and analysis, and my number's right there, 5111. You got, yeah, we can throw out the award search, I guess. Role of antifreeze proteins. Well, maybe we can't. Geez, ORCID 5111. So there's more human genetics. It's become a little more difficult since I've tried to scrutinize it harder, guys. But we're going to get through it, and maybe someday we'll figure it all out. If you move on from that, you have 7193. And 7193 ends up being strain. OH7193. There's one of your strains. Plus, that's my number here, 7193. Neurofibromatosis type 2, genetic and rare diseases. So that number corresponds to that disease, to neurofibromatosis type 2. You got your strain, you got that NM000059 dot BRCA2 colon C7193 C. And it's one of your variants, I believe. Does my no 7193. So you got TRE-TTC3-1 transfer RNA GLUTTC. Atlas of Genetics. You got your strain again. You got Orchid ORCID. Candida genome base. And you got identifier 7193, and they deal with gene mapping. Can't throw that out. You got assessment of genetic diversity and recombination in maize. We've had that before. And so there are several things. I'll provide those links. And oh yeah, taking forever. But But if you move on from that, you have 5567 and 21UR5567 gene, and it is a worm base gene, and it I don't ever figure if I ever figured out if it was a protein coding gene or not. I'm sorry, guys, that these take. Yeah, just stop. It's a worm based gene, all right? Or was it worm based? Yes. I'm not sure if it's protein coding or not, but my internet doesn't want to tell me right now, and we're going on 35 minutes. So, you got that. You got another gene, CAM. You got a gene ID 5567. Protein encoded by this gene. So you got a protein coding gene, PRKACB protein kinase. Defensin gene family. Do I see my number? Yes, 5567. Fly-based gene report, CG5567. United States code. Oh, genetics. Why not? So you got a United States code going on there as well. So some relevant stuff. If you move on from that, you have 1655. And 1655 ended up being non-protein coding RNA again. Among other things, odorant binding protein. You got genetic architecture of Drosophila, whatever. We've had it before. Mutation analysis of chiral inhibitase. Trying to pronounce some of this stuff, guys. Elegans cell death gene CED3. And there's the number. So a new function evolved from gene fusion. Is my number there? Yes, gene fusion. How about another strain? How about RB1655? So you got a few things going on with that, guys. And your last four are going to be 6559, and that is an allele variant. C.6559C. And there's a whole long description of that. But that's not, of course, all you have. You have evolutionary genetics of something I can't pronounce gene. ICD gene. There, I can pronounce that. You got faculty genetics, Yale School of Medicine, which we had before, so it's hard to throw that out. More articles from genetics, PNAS, cancer genetics. Do I see my number? Yes. So cancer genetics again. So you got a few things going on with that, and I will let you guys check it out. I'll provide the links. And now we're going on almost 40 minutes, and I apologize for that. But it's the only way to get through this stuff, really. One search at a time, regardless of how long it takes. And we're through the letters, and we're plugging through the numbers now. And whoever's following along is going to follow along, and for these longer videos. And I think we are getting answers, guys. I think this system is solid. I think it, like I said, I think it can be tweaked later to get more information with some encrypting and stuff. But that'll come later once we get through this really, really long thing on the Bible decode. Maybe we'll try the encryption method with our next bar and grill, Applebee's bar and grill and bar. Maybe we'll try it there. But anyway, guys, apologize for running so long. For now, thanks for all your thumbs up, thumbs down. Thanks for all your comments, leads, feedback, and subscriptions. And thanks for watching, guys. Thanks for bearing with me while I struggle through this information. It is a lot. And especially since I don't know it all, then it becomes difficult for me to present all this information. So that's why you get into taking a while. Also, there's multiple things. So thanks for bearing with us, guys. And for now, you guys have a great rest of your day and have a safe and happy New Year.